I want to start today with a challenge.
Yeah.
I want you to look at your hand. Just, look at the skin. Look at the veins under the skin.
Okay.
If you happen to have a bruise somewhere, look at that too.
It sounds a little bit like a meditation exercise, but knowing you, this is definitely leading somewhere chemical.
It is. Because we walk around in these bodies every single day, and we see all these colors. The red of blood when you get a cut, the yellow of urine, the weird greenish tint of a healing bruise, the brown of, waste.
Right. They're just part of the background noise of being alive.
Exactly. We see them so often they become invisible. We just accept them as the way things are. But they're not just random. There's a reason for every single one of those colors.
You're going to tell me there's a specific reason for the body's palette.
I am. And honestly, when I started reading the source material for today, we're working from Chapter 31, Porphyrins and Bile Pigments, I thought I was in for a really dry, dusty biochemistry lecture. I was expecting to be bored.
And let me guess, you weren't.
Not even close. It turns out the answer to why is blood red and why is Coop brown is actually the same answer. It's all part of the same story.
It absolutely is. It's a single narrative arc. It's the story of one specific family of molecules, and it's a story of creation and destruction.
A life cycle.
A life cycle, how the body struggles to build these molecules, how it uses them to, literally sustain life, and then the messy, very dangerous process of breaking them down and getting rid of them.
And along the way, this one story manages to explain lead poisoning, liver failure, why newborn babies turn yellow, and I'm not making this up, the likely biological origin of vampire and werewolf myths.
It really is the biochemistry of everything. We are talking about heme.
Heme. That's the celebrity molecule for today.
It is the absolute star of the show. Heme is what carries oxygen in your blood. It's the reason complex multicellular life can even exist. Without heme, you and I are not having this conversation.
Right.
But the thing that really struck me looking through this research is just how high stakes this whole manufacturing process is.
That's the exact vibe I got too. It feels like the body is building a bomb. A really useful bomb, but still a bomb. If you build it right, it powers the entire civilization of your body.
Yeah.
But if you drop a screw or miss a single step in the assembly line, the whole thing blows up in your face.
That is a surprisingly accurate analogy. The intermediates, the half-finished versions of heme are actively toxic. They're dangerous. So the body has to run this incredibly tight, incredibly controlled assembly line.
Okay, so that's our mission for this deep dive. We are going to walk that assembly line. We're going to start with the raw materials. We're going to build the heme from scratch, see what happens when the factory breaks. Hello, werewolves. And then we're going to watch the demolition crew take it apart to create the colors of our daily waste.
It's a journey from creation to catabolism.
So let's start with the blueprint. Before we start welding things together in the factory, what are we actually building? The text keeps using this word, porphyrin.
Right. And if you want to visualize a porphyrin, you have to use your imagination a little bit. Forget the complex chemical diagrams you see in textbooks for just a second. Just imagine a ring.
Okay, a ring. Simple enough, like a wedding ring.
A bit bigger. Imagine something like a hula hoop. But this hula hoop isn't one solid piece of plastic. It's actually made of 4 smaller, distinct rings that are all linked together.
Okay, so 4 small rings are kind of holding hands to make one giant super ring.
That's the perfect way to think about it. Those 4 small rings are called pyrrole rings, and they're chemically tied together by these little bridges called methane bridges. So the technical name for this whole super ring structure is a tetrapyrrole.
Tetra for 4.
Tetra for 4. The naming is very straightforward here.
I like it when they make it easy.
Now, this super ring has a very special property. Right in the very center, there's a hole. And that hole is the perfect size and shape to trap a metal ion. It's like a setting in a piece of jewelry that's just waiting for its diamond.
And the diamond that you put in there, the metal, that changes everything.
It changes everything completely. So if you take that ring structure and you put a magnesium ion in the center, do you have any idea what molecule you've just made?
Magnesium. I feel like I should know this from high school biology. It's green.
It's very green. You see it every time you look at the window at a tree or a blade of grass. It's chlorophyll.
Get out. The green stuff in plants, that's a porphyrin.
The very same. Chlorophyll is just a porphyrin ring holding on to a magnesium atom. It's the molecule that eats sunlight.
Okay, that's wild.
Now, take that exact same architecture, that same 4-ring hula hoop, and swap the magnesium out for iron, specifically ferrous iron Fe2+. What do you have?
That has to be heme.
That is heme.
So the fundamental mechanism that allows plants to perform photosynthesis and the one that allows my blood to carry oxygen, they're basically siblings.
They are chemical cousins, absolutely. Nature is incredibly efficient. Once it found a structure that was stable and really good at holding a metal ion, it just kept iterating on it, reusing the same blueprint for different jobs.
That is mind-blowing. It connects us to the grass in a way I hadn't really thought about before.
But heme isn't just for blood, right? I mean, I know hemoglobin is the big one, the protein in red blood cells,
Yep.
but the source material lists a bunch of other uses.
Oh, absolutely. Hemoglobin is the main consumer, for sure. About 85% of the body's heme goes into making hemoglobin, mostly happening in the bone marrow. But heme is a multi-purpose tool.
A Swiss Army knife.
Pretty much. It's also used in myoglobin, which is the molecule that stores oxygen in your muscles so you have a reserve for when you sprint.
Okay.
And it's crucial for the cytochromes.
Cytochromes.
Yeah.
Those are the energy guys in the mitochondria, right?
Exactly right. The electron transport chain. They're the proteins that pass electrons down a line like hot potatoes to generate ATP, our body's energy currency. That whole process relies on the heme iron being able to breathe, taking an electron and then letting it go. It's essential.
So it's not just oxygen transport, it's oxygen storage and energy generation. And even protection. It's a key component of an enzyme called catalase, which is what protects your cells from oxidative stress by breaking down hydrogen peroxide.
So if you can't make heme, you don't just get anemia. Your muscles fail, your basic energy production crashes, and you get poisoned by your own metabolic waste products.
Precisely. It is absolutely fundamental to staying alive.
Okay, so we need this stuff badly. Let's head to the factory floor. How do we build it?
The how is where things get really fascinating from a biological engineering perspective, because this assembly line is, frankly, a logistical nightmare. It takes place in two completely different compartments of the cell.
This detail really stood out to me in the chapter. It starts in one place, leaves, and then has to come back to finish.
Yes, it's a cellular commute. Step one happens inside the mitochondrion.
The power plant of the cell.
Right. Then the intermediate molecule is exported out into the cytosol, which is the watery soup of the cell, for steps 2, 3, 4, and 5. And then for the final 3 steps, the new intermediate has to be transported back into the mitochondrion.
Why? I mean, that seems incredibly inefficient on the surface. Why not just do it all in one place?
The current thinking is that it's all about control and safety. The mitochondrion is the power plant, but it's also a secure vault. By separating the reactants and the enzymes in different locations, the cell can ensure that heme is only made when it's absolutely needed. It prevents accidental production.
Because, as you said earlier, the half-finished versions are dangerous.
Exactly. You don't want half-built bombs just lying around the cell's living room. You keep the dangerous steps cordoned off.
That makes sense. Okay, let's look at step 1 then, the kickoff. We're inside the mitochondrion. What are the raw materials?
We start with 2 surprisingly simple and common ingredients. The first is succinyl-CoA.
Succinyl-CoA. That sounds familiar. That's from the Krebs cycle, isn't it?
It is. It's a key energy metabolite. So the body takes a piece of its energy currency. And the second ingredient is glycine, which is the simplest amino acid you can find. So it's a bit of energy and a bit of protein getting welded together.
And the welder.
Yep.
That's the enzyme.
The enzyme is called ALA synthase, or you'll see it written as ALA. This reaction takes those 2 starting materials and creates the very first unique building block of this pathway, a molecule called delta-aminolevulinate.
Okay. That's a mouthful.
It is. We just call it ALA for short.
ALA. Got it. Now, the notes mention something specific about this welder, this ALA enzyme. It needs a battery to work.
It needs a cofactor, yes. And that cofactor is pyridoxal phosphate.
Which is just a fancy name for vitamin B6.
Correct. And this is a huge clinical pearl. If a patient is malnourished or has a terrible diet and is deficient in vitamin B6, this very first enzyme, ALA, cannot work. It can't weld the glycine and succinyl-CoA together.
So the entire heme synthesis pathway stops before it even starts.
Completely. And you get a very specific kind of anemia from that called sideroblastic anemia, all from a simple vitamin deficiency.
It's amazing how fragile we are sometimes. Okay, so assuming we ate our vitamins, we have ALA. The text emphasizes that this is the rate-limiting step, right?
Yes, this is the throttle. This is the single most important control point for the entire 8-step process. The body regulates this enzyme, ALAS, incredibly tightly. If the cell needs more heme, it hits the gas right here. If it has enough, it slams on the brakes right here.
And everything else downstream just has to keep up with the leader.
Exactly. It sets the pace for the whole factory.
Okay, so we've made ALA, We're still inside the mitochondrion. Now it's time for that commute. We go out into the cytosol.
Right. Now we need to start building that ring structure we talked about. So step 2 is to take 2 of those ALA molecules and condense them together to form a pyrrole. Remember, we need 4 of these little rings to eventually make the big super ring.
So we're building the individual subunits,
Yeah.
the pearls for our necklace.
Precisely. This first pyrrole ring molecule is called porphobilinogen, or just PBG, and the enzyme that does this is called ALA dehydratase.
And here's where the story takes a really dark turn. I'm looking at the toxicology alert section in the notes. This is where lid comes in.
Yeah, this is a classic board exam question, and for good reason. This enzyme, ALA dehydratase, is a metalloenzyme. It requires a zinc atom to function. It holds on to that zinc atom to do its job correctly.
But zinc has a bully, a chemical bully.
Lead is a heavy metal bully. If you ingest lead from old paint chips, contaminated water, whatever the source, the lead gets into your cells, finds this enzyme, and it physically kicks the zinc out. It takes its seat in the enzyme.
And I'm guessing lead is a terrible employee and it doesn't do the work.
Lead is a terrible worker. It just sits there and does nothing. It completely paralyzes the enzyme.
Okay, so let's trace the consequences of that. You have lead poisoning. This enzyme, ALA dehydratase, stops working. That means you can't make PBG. If you can't make PBG, you can't make heme.
Which means you get anemia. That's symptom number 1 of lead poisoning. But then you have to think about what happens before the blockage.
The assembly line backs up. You get a huge traffic jam.
A massive traffic jam. The stuff that comes before the broken enzyme, the ALA, it just piles up. You get massive amounts of ALA accumulating in the blood and tissues.
And that's bad.
That's very bad. Because it turns out ALA has a chemical structure that looks suspiciously similar to a major inhibitory neurotransmitter in your brain called GABA.
Oh, no.
Oh, yes. So this flood of ALA crosses the blood-brain barrier, it gets into your nervous system, it starts messing with your neural signaling, pretending to be a neurotransmitter. It causes severe neurotoxicity.
So that's why lead poisoning isn't just, I'm pale and tired from anemia. It's the behavioral changes, the developmental delays in kids, it's actual brain damage.
It's because a backlog of unfinished blood components is essentially frying the neural circuits.
That is terrifying. Just one metal ion swapping places, and it can ruin your mind.
Biology is a game of inches. The details matter.
Okay, let's assume we are lead-free for the rest of this. We have our PBG molecules. We need 4 of them to make the big ring.
Correct. So step 3 involves another enzyme that gathers up 4 of those PBG molecules and strings them together head to tail into a line, a linear chain.
A string of 4 pearls.
A string of 4 pearls. The technical name is hydroxymethylbalane.
I'm sorry, hydroxy what?
Hydroxymethylbalane.
That sounds like something on the back of a shampoo bottle that you're not supposed to get in your eyes.
It does, and honestly, it doesn't last long enough to be a shampoo ingredient. It's very unstable. Because now we face the most critical, most geometrically challenging step of the entire process. Step 4, the ring closure.
The outline calls this the isomer fork. This sounds important.
This is a detail that separates the casual students from the real experts. So we have this string of 4 rings. We need to curl it up and connect the ends to make a circle. But it's not that simple.
Why not?
Because these rings have little decorations on them, side chains. And they come in 2 flavors. Short ones called acetate and long ones called propionate.
Okay, so like a pattern on the pearls. Short, long, short, long.
Exactly. Let's call them A and P for short. So the natural string is built as AP, 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 a nice repeating pattern. Now, if you just let that string curl up and connect the ends naturally, spontaneously,
You get a perfectly symmetrical ring.
yeah,
AP, 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 all the way around.
you do. We call this the type I isomer.
And symmetry is usually good in nature, right? We like symmetry. It's efficient.
Usually. But in heme synthesis, symmetry is trash.
Crash.
Biological garbage. Type I porphyrins, the symmetrical ones, cannot be used to make heme. They are a metabolic dead end. The enzymes further down the assembly line literally cannot hold them. They don't fit into the active site.
So we built a key, but the teeth are all cut exactly the same, and the lock needs a key with different sized teeth.
That's a perfect analogy. So nature evolved a specific enzyme. It's called uroporphyrinogen III synthetase that does something incredibly tricky and elegant. It grabs that linear string of 4 pearls, and right before it snaps it shut into a ring, it flips the final pearl.
It flips it around.
It reverses it. So instead of the string closing as AP, 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 it closes as AP, AP, APA.
It breaks the pattern on purpose.
It creates asymmetry. This is called the type 3 isomer. And this asymmetrical, slightly wonky-looking ring is the only one that works for life.
That is just bizarre. Is there a reason why?
It's likely due to the final three-dimensional shape that's required to fit snugly into the pockets of the later enzymes and eventually into the hemoglobin protein itself.
It's like buttoning a shirt. If you
Yeah.
button it perfectly straight, it might look neat, but it won't fit your body right. You have to offset one side for it to work.
That's the analogy from the outline, and it's a good one. The buttons on the porphyrin shirt have to be misaligned for the shirt to fit the body of the protein.
So if this special flipping enzyme is broken due to a genetic defect,
Then the chain just closes spontaneously without the flip. You don't make the useful type 3. Instead, you get piles and piles of type I, the symmetric, useless garbage.
and since the body can't use it or break it down easily, it just accumulates.
It accumulates, and I'm guessing that's very bad.
Oh, it's the basis for one of the most disfiguring and myth-inspiring diseases in all of medicine. But we'll get there. Let's stick a pin in the garbage ring for now.
Okay.
So we have our correctly flipped asymmetrical type 3 ring. We're still in the cytosol. Now the outline says we have some trimming and modifications to do.
Yes. Steps 5 through 7 are all about refinement. Think of the molecule at this stage as a rough draft. It's bulky, it has these big water-loving side chains, the acetate and propionate groups sticking out, but he ultimately needs to fit into hydrophobic, sort of oily pockets and proteins.
So you need to sleek it down, make it less water-friendly and more oil-friendly.
We need to make it more aerodynamic, chemically speaking. So we start by chopping off carbon dioxide molecules. This is called decarboxylation. We turn the bulky acetate groups into much smaller methyl groups.
Okay.
Then the new molecule, which is called coproporphyrinogen III, re-enters the mitochondrion for the final touches.
Back to the vault.
Back to the vault. Inside, other enzymes go to work on those long propionate groups. They oxidize them and turn them into vinyl groups.
And vinyl groups are greasy.
They are hydrophobic, they're sleek. We are essentially stripping away the water-loving parts and replacing them with parts that prefer an oily environment. We're tailoring the molecule for its final home.
Got it. We've trimmed the fat. We are back in the mitochondrion. We have reached the grand finale, step 8.
The stage is now perfectly set. We have a molecule called protoporphyrin III. It is a flat, rigid, intensely colored disc with a hole right in the middle, just waiting.
Enter the hero of the story, the metal.
Enter the final enzyme, ferrochelatase. This enzyme is the master craftsman. It grabs a ferrous iron atom, an F2+ ion, and with incredible precision, it forcibly inserts it right into the center of that ring.
Click. The diamond is set.
And just like that, the molecule comes alive. It's no longer just a porphyrin, it becomes him.
And of course, I have to ask, is our old villain, lead, lurking here at the finish line too?
Sadly, yes. Lead is a persistent villain in this story. It also inhibits ferrochelatase. So if you have significant lead poisoning, you are blocked at the very beginning of the pathway with ALA dehydratase, and you're blocked again at the very end with ferrochelatase. It's a devastating double tap.
Wow. That explains why the anemia from lead poisoning is so severe. You're fighting a war on two fronts. You can't start and you can't finish.
Exactly.
Okay, before we move on to what happens when this process goes wrong genetically, I want to clear up something about color. The title of our deep dive is The Colors of Life. But you mentioned a minute ago that the intermediates are colorless. How does that work?
This is a massive point of confusion for students, and it's a fantastic question. The molecules we were building in the middle of the pathway, uroporphyrinogen, coproporphyrinogen, notice that common suffix, -ogen. In biochemistry, otogen usually means precursor or potential. Think of pepsinogen becoming pepsin. In this specific case, it means the molecules are chemically reduced. They have extra hydrogen atoms attached. The system of double bonds around the ring isn't fully connected yet.
It's not a complete circuit, so light just passes right through them.
Exactly. If you had a beaker full of pure uroporphyrinogen, it would look just like water. It's colorless.
Yeah.
But if you leave that beaker on the lab bench, exposed to air and light,
Oxygen gets to it.
oxygen is an electron thief. It comes along and steals those extra hydrogens. The molecule becomes oxidized, it loses the ogen, and is now just a porphyrin, porphyrin.
And now the circuit is complete.
The double bonds connect all the way around the ring. We call that a conjugated system, and that resonance is what allows it to absorb light.
Absorbing light means color.
Deep, intense color, usually a dark red or purple. And it's not just color, it's also fluorescence.
They glow, like under a black light.
If you shine ultraviolet light on oxidized porphyrins, they glow a brilliant almost hellish red. It's incredibly distinctive.
That is so cool.
It is. And it's not just a cool party trick. We actually weaponize this exact property to fight certain types of cancer.
Wait, what? We use these glowing blood precursors to fight cancer. How?
It's a treatment called photodynamic therapy, or PDT. Here's the trick. Cancer cells are greedy. They're growing and dividing so fast, so they have a very high metabolism. They just eat everything in sight.
Okay.
If you inject a patient with a solution of porphyrins, the tumor cells will gobble them up much, much faster than the surrounding healthy cells.
So you've selectively loaded the tumor with these light-sensitive molecules.
Right. Then, a day or two later, you take a laser, specifically an argon laser, that's tuned to a wavelength of about 400 nanometers, which is the peak absorption for porphyrins. We call it the SORT band.
Right.
You shine that specific wavelength of laser light directly onto the tumor.
And the porphyrins inside the cancer cells absorb that laser energy.
They absorb it like a sponge. They get incredibly excited, chemically speaking, and then they have to release that energy somehow. They do it by transferring the energy to nearby oxygen molecules, creating reactive oxygen species, mainly singlet oxygen.
Free radicals.
Basically, you're creating millions of tiny molecular grenades. The porphyrins inside the cancer cells detonate, releasing this oxidative burst that destroys the tumor from the inside out, while leaving most of the healthy tissue unharmed.
That is something out of science fiction. We are turning the building blocks of our own blood into laser-activated depth charges to kill cancer.
It's one of the most elegant and targeted therapies we have, but it all relies on that fundamental property. Porphyrins react violently to light, and that brings us to the dark side of the story.
The porphyrias.
The porphyrias. These are the diseases, mostly genetic, where the factory breaks down.
And there are a lot of them, right? The table in the source material lists one for pretty much every enzyme.
There's a specific disease for almost every single step in the chain. But to keep things simple for diagnosis, we divide them into two big buckets: acute porphyrias and cutaneous porphyrias.
Acute meaning sudden and painful, and cutaneous meaning related to the skin.
That's it. And it all depends on where in the assembly line the clog is. If the clog is early in the chain, before the ring is even formed, you get a buildup of those early precursors, ALA and PBG.
The ones you said were neurotoxic.
Exactly. So these patients don't have skin problems because those molecules don't react to light. They have brain and gut problems. They suffer from acute porphyria.
And this brings us to one of the most famous patients in history.
King George III of England, the Mad King who, lost the American colonies.
I've seen the play. I've seen the movie, The Madness of King George. But what was actually happening inside his body?
Historians and medical doctors now widely believe he had a genetic disease called acute intermittent porphyria. He suffered from these intermittent, unpredictable attacks of excruciating abdominal pain.
Which is the ALA and PBG affecting the nerves in his gut.
Right. But he also had the famous madness, the hallucinations, the severe paranoia, the episodes of talking nonstop for hours until he frothed at the mouth. That was the neurotoxicity. And the smoking gun.
What was it?
His own doctors, back in the 1700s, recorded in their notes that during these attacks, his urine was bluish or turned a dark port wine red upon standing.
That's the precursor molecules spilling into the urine and then oxidizing in the chamber pot.
Exactly. The poor man wasn't insane in the way they thought. He had a broken enzyme. And if you think about the triggers for these attacks, it makes even more sense. Porphyria attacks are often triggered by things that ramp up the heme factory, that push the accelerator.
Like what kind of things?
Stress is a big one, alcohol, starvation, and, critically, certain drugs. This brings us back to the regulation part we briefly skipped over, the P450 connection.
Oh, right. Okay. So the liver uses heme for detox.
Right. The liver uses a huge amount of HAM to make a family of enzymes called cytochrome P450. This is your body's main detoxification system for drugs, toxins, everything.
So if you take a drug, the liver says, I need to break this down.
Exactly. It says we need to break this down, build more P450 enzymes now, and to build more P450, it has to consume its stored HAM.
It drains the heme reserves. Now, I remember the throttle. The first enzyme, ALAS1, is controlled by heme levels. If heme is low, ALAS1 speeds way up.
Precisely. The drug lowers the heme level, which acts as a signal. The brake comes off LAS1, the throttle gets ripped wide open, and the factory starts churning out precursors at 1,000% speed to try and make more heme.
But if you have a genetic clog further down the line...
You have just slammed the accelerator on a car that has a banana stuck in its tailpipe. The pressure builds up behind the clog, the neurotoxic precursors flood the entire system, and you go mad.
So the very doctors giving him medicines, probably barbiturates to try and calm him down, were actually fueling the chemical fire that was driving him crazy.
It's a tragic, iatrogenic feedback loop. They were making him worse.
That is just, it's heavy. Okay, so that's the acute side of the coin. Madness and pain from the early precursors. What about the cutaneous side? The skin problems.
This happens when the enzyme clog is later in the chain after the ring structure has already been formed and become a porphyrin. You get a buildup of these colored, light-sensitive porphyrins in the blood.
And these ones don't go to the brain.
No. For the most part, they are hydrophobic, they're greasy, they don't like the blood, so they deposit in fatty tissues, especially the skin. And now, you have to remember the cancer laser.
They react to light.
Viciously.
Yeah.
So these patients walk out into the sunlight. The photons in sunlight hit the porphyrins that are embedded in their skin cells. The porphyrins absorb the energy, get excited, generate those free radicals, and literally cause the cells to explode.
So the sun burns them from the inside out.
It's not just a sunburn. We're talking severe blistering. The skin can shear off. It leads to terrible scarring and disfigurement.
And this. This is what leads us to the mythical creatures in the textbook.
This is it. The most extreme example is a rare disease called congenital erythropoietic porphyria. It's a mouthful. But this is the disease where that special flipping enzyme is broken. So you make tons and tons of that useless symmetric type I porphyrin.
The garbage ring.
Right. It can't be used, so it just saturates the body. The patient has the most severe photosensitivity imaginable. They can only come out at night.
Okay, so that's vampire check number 1, nocturnal.
The scarring on the face from repeated sun exposure can be so bad that it erodes the cartilage of the nose and ears. The gums can recede and pull back from the teeth, making the teeth look abnormally long, like fangs.
Fangs, vampire check number 2.
And the porphyrins themselves, being colored, deposit in the dentin of the teeth. Under normal light, the teeth might look reddish-brown, but under a UV light, they fluoresce a brilliant glowing red.
Glowing red teeth. Okay, that's terrifying.
Oh. And finally, in some cases, the body tries to protect the damaged skin by growing a fine, downy layer of hair all over the face and arms. It's a condition called hypertrichosis.
So we have a nocturnal person with fangs, glowing red teeth, and a hairy face who screams in agony if sunlight touches them.
It is very, very difficult to look at that complete clinical presentation and not see the biological origin of the werewolf and vampire legends from medieval Europe.
It makes you wonder how many people who were condemned as monsters throughout history were just people suffering from a single missing enzyme.
I would guess most of them.
Wow. Okay. We have covered creation and the disasters of creation. Now we have to clean up the mess. Section 5. The death of heme.
And this is a monumental job. Your body has to dispose of about 200 billion old red blood cells every single day.
200 billion. That number doesn't even feel real. It's astronomical.
It's staggering. That turnover releases about 6 grams of hemoglobin daily that has to be dealt with. Now, the body is very, very good at recycling. It takes the protein part, the globin, and breaks it down into amino acids to reuse for new proteins.
Good.
It very carefully extracts the iron atom and puts it back into the body's iron pool to be used again.
Also good. But what about the ring, the porphyrin part?
The ring is toxic waste. It's dangerous. You can't reuse it. You have to take it apart and throw it away.
And this demolition work happens in the spleen.
The spleen and the liver, mostly. In specialized cells of the reticuloendothelial system, this is the graveyard of the red blood cells, the enzyme that swings the molecular wrecking ball is called heme oxygenase.
And this enzyme does something that genuinely shocked me when I read it in the notes.
Mm-hmm.
It produces a poison.
It's one of the weirdest and coolest facts in all of human biology. Heme oxygenase cracks open the porphyrin ring. To do that, it uses oxygen. And one of the byproducts of this chemical reaction is carbon monoxide, COO.
Carbon monoxide. The colorless, odorless gas that kills you if you leave your car running in a closed garage.
The very same. We produce it inside our own bodies, on purpose. It is the only known endogenous source of carbon monoxide in biology.
Why? I mean, isn't it a poison? Why would we make it?
In high doses from an external source, yes, it's a poison because it competes with oxygen for binding to hemoglobin. But in these tiny, tiny doses produced locally in tissues, CO actually acts as a gasotransmitter, a signaling molecule. It tells nearby blood vessels to dilate. It's anti-inflammatory.
So we use a poison as a tool.
We do. You are literally exhaling a tiny puff of carbon monoxide with every breath right now because your spleen is crushing old blood cells.
That is wild. Absolutely wild. Okay, so we crack the ring open with heme oxygenase.
What's left? When you open the ring, it's no longer a circle, it's a linear chain. And the color changes dramatically. The deep red of heme becomes the bright green of a molecule called biliverdin. Green. This is interesting. If you look at birds or reptiles, their droppings are often green and white. The green part is biliverdin. They just excrete it directly at that stage.
But we aren't birds. We take it a step further.
We do. We have another enzyme called biliverdin reductase. It takes the green biliverdin and through a reduction reaction, it turns it into the yellow-orange pigment, bilirubin.
And the host notes here have a great visualization for this whole sequence. The life cycle of a bruise.
It's the perfect real-world timeline. Think about a bad shiner. You get hit in the eye.
Okay, I got punched in the eye.
Day 1, it's dark purple, maybe reddish black. That's the intact hem from all the blood that's spilled under your skin.
Right.
Day 3 or 4, you'll notice the edges start to turn a sickly green color. That's your local macrophage cells firing up their hem oxygenase. They're making biliverdin.
That's the green phase.
Then, around day 7 or so, the bruise turns that weird, ugly, yellow-brown color as it's healing. That is biliverdin reductase doing its job. It's making bilirubin.
So a bruise is just a color-coded progress bar of your enzymes cleaning up a mess.
Exactly. But now we have a big problem. We've made bilirubin. It's yellow. But chemically, it is a nightmare for transport in the body.
Why is that?
It is incredibly hydrophobic. It's greasy. It's oily. It hates water. And your blood is mostly water. If you just dumped it into the bloodstream, it would clump up and stick to your vessel walls. It wouldn't flow.
So it needs a chaperone. Can't travel alone.
It needs a taxi service. And that taxi is a protein called albumin. Albumin is the most common protein in your blood plasma, and one of its main jobs is to act as a carrier for hydrophobic molecules like this. It grabs the greasy bilirubin and wraps it up so it can float safely through the blood to the liver.
This gives us our first major medical definition for jaundice, right?
Right. When bilirubin is attached to albumin on its way from the spleen to the liver, we call it unconjugated bilirubin. You'll also see it called indirect bilirubin on lab reports.
Indirect because it hasn't hit the liver's processing plant yet.
Correct. So the albumin taxi drops it off at the liver. The liver cells, the hepatocytes, act like a customs checkpoint. They take the bilirubin off the albumin and bring it inside the cell. Now, the liver has to solve that grease problem permanently. It needs to make this molecule water-soluble so it can be dumped into the bile and excreted.
How does it do that? How do you make something greasy water-soluble?
You attach a big water-loving sticker to it. Specifically, it attaches a sugar-like molecule called glucuronic acid.
It frosts the greasy donut to make it palatable.
That's one way to put it. The enzyme that does this is a mouthful.
Yeah.
UDP, glucuronosyl transferase, we'll just call it UDT. It slaps 2 of these sugar acid molecules onto the bilirubin. Now, the resulting molecule is polar. It loves water.
And we call this new water-soluble version
Conjugated bilirubin, or, on a lab report, direct bilirubin.
Okay, I want to pause here because this is the absolute key to understanding any lab report for a patient with jaundice. Unconjugated or indirect means it's the greasy pre-liver form. Conjugated or direct means it's the water-soluble post-liver form.
That is the perfect summary. Once it's conjugated, the liver cell actively pumps it into the tiny bile ducts using a specialized pump protein called MO8, and from there, it flows down into the gallbladder and then into the intestine.
And here is where I feel like the story gets underappreciated. The intestine. We usually think, okay, the waste is in the gut, the story's over, it's gone, but it's not gone yet.
No, not at all. This is where your microbiome, your gut bacteria, enter the chat. You have billions and billions of bacteria in your gut. They see this conjugated bilirubin arriving from the liver, carrying those delicious sugar acid stickers.
And bacteria love sugar.
They absolutely love it. They have their own enzymes called beta-glucuronidases that just rip those sugar stickers right off to eat them.
Rude. So now the bilirubin is unconjugated again.
Yes, but then the bacteria go even further. They metabolize the bilirubin itself, chemically modifying it into a group of completely colorless molecules called urobilinogens.
Colorless. So it went from red heme to green biliverdin to yellow bilirubin and now to colorless urobilinogens.
Briefly, yes. Now, this urobilinogen has 3 possible fates. This is the choose your own adventure part of the waste disposal story.
Okay, adventure number 1. What's behind door number 1?
Recycling. About 20% of the urobilinogen that's made in the gut is reabsorbed from the gut back into the bloodstream. It goes back to the liver, and the liver just pulls it out of the blood and puts it right back into the bile.
It's the enterohepatic cycle.
That's it. The body is thrifty. It keeps that cycle going to a certain extent.
Okay, adventure number 2.
Some of that reabsorbed urobilinogen slips past the liver's cleanup crew and stays in the blood. It gets to the kidneys. The kidneys filter it out of the blood and into the urine. In the urine, it gets exposed to air and oxygen, and it oxidizes into a yellow-colored molecule called urobilin.
And that is why urine is yellow.
That is precisely why urine is yellow. It is literally the oxidized rust from your old, broken-down red blood cells.
Amazing. Okay, adventure number 3.
This is the fate for the bulk of it. It just stays in the colon. The bacteria in the large intestine keep chewing on it, modifying it further. It oxidizes and turns a deep brown color. It becomes a molecule called stercobilin.
And that is why feces are brown.
That's it. The entire color palette of our waste products is dictated by the breakdown of heme, which also means if you have no bile flow,
your poop would be white or pale.
Exactly. We call it a colic or clay-colored stool. It's a classic sign that a patient's bile duct is blocked. The pigment, the bilirubin, never gets to the gut, so the paint never gets applied to the final product.
It's amazing. The color of everything that comes out of us is determined by this one specific chain of breakdown.
It is. And when that yellow color ends up in the wrong place, like in the skin or the whites of the eyes, we call it jaundice.
Section 8. Jaundice, or hyperbilirubinemia. I feel like we are ready to be medical detectives now. We know the whole pathway from start to finish.
That's exactly how doctors use this knowledge. Jaundice itself is just a symptom. Your skin is yellow. The real question is why. And the lab values, specifically the direct versus indirect bilirubin, tell you the story of where the plumbing is broken.
Okay, let's play the game. Case number 1. A patient comes into the ER and they're yellow. You run their labs and they show a very high indirect, the unconjugated bilirubin. The direct bilirubin is normal. What's happening?
Okay, high indirect means the liver hasn't touched it yet. That implies the liver itself is probably working just fine, but it's being completely overwhelmed. The delivery trucks are dumping way too much raw material at the factory door.
So massive red blood cell destruction.
Exactly, hemolysis. This could be from malaria, a sickle cell crisis, a bad blood transfusion reaction. The body is destroying red blood cells so fast that the waste management system, the liver, just can't keep up. We call this prehepatic jaundice. The problem is before the liver.
Makes sense. Okay, case number 2. A patient is yellow. They tell you their urine is dark, like cola, and their stool has been pale, almost white, like clay. You run their labs, and they have a very high chemistry. Direct, the conjugated bilirubin.
Okay, high direct tells you the liver did its job. It received the bilirubin, it put the water-soluble stickers on it, but it couldn't get it out. The exit is blocked. And the pale stool confirms it, the pigment isn't getting to the gut.
So it's a blockage in the pipes after the liver.
Posthepatic jaundice, or obstructive jaundice. A gallstone could be stuck in the common bile duct, or maybe a tumor in the pancreas is pressing on the tube. And that dark urine is the water-soluble conjugated bilirubin backing up, overflowing into the blood, and being filtered out by the kidneys.
Got it. In case 3, a mix. Both the indirect and the direct bilirubin levels are high.
That's usually hepatic jaundice. The liver itself is sick. Think hepatitis from a virus or cirrhosis from alcohol. The liver cells themselves are damaged and dying. They can't conjugate fast enough, so the indirect goes up. And the liver's architecture is ruined, so the conjugated bilirubin that they do manage to make just leaks back out into the blood instead of going into the bile, so the direct goes up too.
It's just logic. It's plumbing logic applied to the body.
It really is. You just have to follow the flow of the pigment.
Before we wrap up, we have to talk about the most common jaundice of all. The one we see every day in hospitals. Babies.
Neonatal jaundice, or physiologic jaundice of the newborn. It happens in something like 60% of all full-term newborn.
Why? Is their liver broken?
Not broken, just immature. That key enzyme, the UGT enzyme that adds the sugar sticker, it takes a few days to a few weeks to really ramp up to full capacity after birth. But at the same time, the baby's body is rapidly breaking down all of its old fetal red blood cells.
So you have a massive influx of raw material and a very slow factory.
A perfect storm. The result is a high level of unconjugated, greasy bilirubin.
Is it dangerous? Or is it just a cosmetic thing that they'll grow out of?
It can be extremely dangerous. Remember that unconjugated bilirubin is greasy. It loves fat. And what is the brain mostly made of?
Fat. Oh, no.
In adults, we have a very strong blood-brain barrier that keeps it out. But in newborns, that barrier is still leaky. If the bilirubin levels get too high, that greasy yellow pigment can cross into the brain and it literally stains the basal ganglia yellow.
That sounds horrific.
It's called kernicterus, and it causes permanent, irreversible brain damage, cerebral palsy, hearing loss, developmental delays. It's an absolute catastrophe if it happens.
But we see yellow babies all the time, and they turn out fine. How do we treat it? How do we fix it?
We use light. Again, this whole story is about light.
The famous blue lights, phototherapy.
Phototherapy. But this is a different mechanism from the cancer laser. We put the baby under a special bank of blue lights. The light energy from those lamps shines on the baby's skin, penetrates a little way, and it hits the bilirubin molecule that's sitting there, and it twists it.
It twists a molecule.
It causes a structural change, a photosomerization. It doesn't add or remove anything, it just changes the shape. It turns the toxic, greasy bilirubin into new shapes called photoisomers. And these new isomers are, weirdly enough, water-soluble without needing the liver to add the sugar sticker.
The baby can just pee them out directly.
The baby can excrete them directly into the bile and urine. The light does the liver's job for it. It creates a bypass route for excretion. It is a miraculous, completely non-invasive cure for a potentially fatal condition.
That is just. It's elegant. Biology breaks it, and physics fixes it.
It's a perfect partnership.
We have covered a massive amount of ground today. From the chemical rings of chlorophyll to the madness of kings, from the green of a healing bruise to the blue light of the newborn nursery.
It's a very dense chapter. But if you can just remember the core story, build the ring, flip the ring, fill the ring with iron, and then break the ring, you understand the whole thing.
And I think it fundamentally changes how you see your own body. We aren't just these static sculptures walking around. We are these churning, dynamic factories, constantly building explosives and then safely dismantling them every second of every day.
And doing it with a level of precision that makes our best industrial chemical plants look like clumsy children playing with Legos.
I want to leave the listener with that thought we touched on briefly earlier, the carbon monoxide thing.
Yeah.
We spent decades, a century, thinking carbon monoxide was just a poison, an industrial waste product. Then we find out our spleen makes it on purpose as an essential tool.
Yeah.
It makes me wonder, what other toxins or waste products are we trying to eliminate from our bodies right now that are actually secret keys to our health that we just don't understand the function of yet?
That is the ultimate question in physiology and medicine. One man's poison is nature's essential signaling molecule.
It's something to chew on. Thanks for taking this deep dive with us.
Always a pleasure.